The Duke Superfund Community Engagement Corps held its annual Community Advisory Board meeting virtually on July 17, 2020. We were fortunate to have presentations from 2 Superfund trainees and 1 investigator from the Nicholas School of the Environment to share their work with our board. The researchers also participated in discussions on topics related to our Community Engagement Corps throughout the day.
I am a Superfund trainee. I'm a PhD student working with Joel Meyer in Project 3, which is trying to understand what are the long-term effects of low-level exposures of chemicals. And specifically, my question that I'm going to be talking a little bit about today was, what are the long-term effects of persistent exposures of chemicals on DNA? And I'm going to be introducing you to a few critters I work with in the lab. Today, I'm mostly going to be telling you a story about the freshwater crustacean Daphnia. And yeah, get started. So I don't need to tell all of you, but our health, ecosystem health, are interconnected with the health of our environment. And myself and other researchers have recently been interested in trying to get at this question: what are the effects of chemicals on DNA? And so can we use DNA kind of as a fingerprint to understand what different exposures have occurred in different organisms, including humans? And so today I'll briefly give some background and an overview of my research question. I'll describe the methods of one experiment I did and some of the results. And then most importantly, which I'd love feedback on to continue this discussion, is how does my research apply to community health? So you may be wondering why DNA, as you might have learned a long, long time ago in biology class, but DNA is the blueprint for all of life. 4 different DNA molecules are responsible for storing and passing on across generations all the information that is needed to regulate how organisms grow, develop, and function. And this is encoded in what are called genes. So each gene is responsible for building a specific protein. For example, there's a gene that when signaled, such as exposure to a pathogen, such as a virus, a protein is built, like an antibody, to defend ourselves against this virus. So not only does DNA and specific genes account for all the different proteins that are made, but the different sequences of DNA are responsible for all the variation in life that we observe. So differences in sequences account for biodiversity, but also differences in DNA sequence account for all the diversity, even within a species. So for example, in this alpine wildflower, there's a lot of variation that we can see. And among ourselves even, there's a lot of changes in our DNA that make us different from one another. And the ultimate source of this genetic variation is called a mutation. And so, for example, on this DNA sequence on the left, where we have what was a C is now a T, and this is called a C to T mutation. And what can happen sometimes is a mutation in the DNA might result in the change in the structure and potentially the function of a protein, which is how we can see our variation. And so most mutations happen randomly and aren't problematic, but we do know that sometimes chemical exposures can cause different mutations. So for example, ultraviolet radiation can cause damage to DNA and skin cells, and this can maybe cause a mutation that results in skin cancer. Another example that we know of is that polycyclic aromatic hydrocarbons that are found in cigarette smoke can cause damage to DNA in the lung tissues and potentially result in lung cancer. But we actually don't know a lot about a lot of different chemicals and how they might cause damage to DNA and ultimately mutations that might result in different health implications. And so what I'm interested in doing is searching for mutational signatures that might be indicative of specific chemical exposures and the effects of the chemical on the change in the DNA. And so just like our own unique signatures provide our own identity, it's possible that when we sequence DNA, the changes that we see in the DNA might be a signature, might show a signature that is caused by a specific chemical. And so, for example, I've outlined in these red boxes here, these are two very distinct mutational signatures where we see this C to T mutation that I mentioned earlier, whereas in this sample that was exposed to a different chemical, we see a C to T mutation. And so how can this be used to educate ourselves on what exposures, what an organism was exposed to and the potential effects of this mutation? And so I'm going to describe to you one experiment that I've done and some results at how we're trying to answer this question of the effects of chemicals on DNA. And the chemical that I'm interested in is cadmium. It's a ubiquitous pollutant. It's released in industrial combustion and emissions, electronic waste sites, and also mining drainage. And because of all these various sources, exposure risk is pretty high in many different forms, so air and water and even soil. And so in the lab, I'm using this chemical cadmium to look at effects of cadmium on mutations. And so I did an experiment in this organism called Daphnia. It's about the size of the letter D on your keyboard if you look down right now. It's very small. It's found in freshwater lakes all over the world. It's been very well studied by ecologists and toxicologists. And so in the lab, I took one individual, allowed the individual to reproduce clonally to produce genetically identical offspring. I then randomly selected one individual and passaged that individual into its own clean beaker of water. I allowed that one to grow up and reproduce, randomly selected one of their offspring, passaged on to the next generation, and repeated this for many, many generations. And the premise of this experiment is such that if a mutation arises, so if there's a change in the sequence of DNA at any of these time points, it is then stuck in that beaker, in that individual, and is passage on each consecutive generation. So at the end, you've accumulated a lot of mutations that we then sequence to measure the changes in the DNA sequence compared to the starting individual. And so what we did in parallel was repeated this experiment, but instead of in clean lab water, we used a concentration of cadmium that is relevant to pollution found in lakes and aquatic environments. And so after repeating these experiments, we sequenced the DNA after 3,000 and 12,000 generations of exposure. So this is a very, very long-term experiment that we obviously couldn't do in humans, but we're using this organism to try to understand the effects of cadmium on mutations. So we then compared the number of mutations in the cadmium experiment to the control experiment. And so these are the mutational signatures that I described earlier. So on the left, we have a graph showing the number of mutations that we added up after 3,000 generations. And so when we look at the cadmium Daphnia populations, we see that there's an increase in this GC to AT. So we have this specific mutational signature. Interestingly, after 12,000 generations, we see a different mutational signature. We see an increase of AT to GC mutations in the cadmium compared to the control animals. So why didn't we see a clear signature caused by cadmium? Well, there's a few hypotheses that I have that I'm working on in the lab. So one would be maybe the DNA sequencing isn't good enough to detect small changes in the DNA. So I'm looking at a newer, more advanced sequencing technology in two different organisms, killifish and C. elegans. And then secondly, maybe there's a mechanism that cells have to get rid of the DNA damage caused by cadmium that we don't know about yet. And I'm using C. elegans in Dr. Meyer's lab again to try to answer some of these questions. So how does this apply to community health? Well, ideally, I'd like to link my experiment in the lab to a relevant story where we do see a lot of cadmium pollution caused by industrial contamination. And one place where this has been of significant concern is in Sudbury, Ontario, where there's been over 130 years of industrial mining that's released a lot of cadmium and other heavy metal pollution and has caused a lot of contamination in surrounding lakes, but also has decimated the whole landscape from acid rain, resulting in Sudbury to looking like a moonscape. But remarkably, there are populations of Daphnia, the same organism that I used in my lab experiments, that have become tolerant to cadmium, and we can find them in these very polluted lakes in this region. And so our next step is to try to understand what's going on in these organisms. How can that maybe provide some insight into the potential changes and health implications in people that might be living in this area or other areas exposed to heavy metal pollution. And so I don't want to leave on a negative note. There have been some regreening efforts in Sudbury over the past 4 decades, but unfortunately, we still don't know these long-term health effects that might be associated with those living near mining and heavy metal pollution. And there's been recent efforts to try to address some of these health disparities that are occurring in the Sudbury region. And so my goal would be to try to use this model and these questions. Can we look at changes in DNA as a fingerprint to what other chemicals are out there in specific regions in humans, but also in other organisms? This is potentially even relevant to North Carolina, where we see a lot of legacy pollutants in former industrial sites around the state. So with that, I'd like to thank the Meyer Lab and everyone in it, and specifically Dr. Joe Shaw, who I collaborated with on the Daphnia project, and of course the Superfund program for all of their support over the past 5 years, and the Nicholas School. So with that, thank you, and I'm happy to answer any questions if I have time.